they have lots of problems as they grow. The people showed in this particular setting that this occurs due to loss of GR, this is glucocorticoid receptors in the hippocampus. That gene gets turned off only in the babies who are brought up by mothers who do not care about their babies, or poor care, and this gene turns off, and that obviously results in persistent abnormal behavior, and that is then inherited in their babies. So when those babies make babies in the next generation, those genes get inherited. GR is actually shut down, and that gets inherited. There's a Nature Neuroscience paper. I'll send it to you guys. Read it if you want. And then there's a lot more work done on the space. They show also the ER, etc. is also there. So what does that tell you, right? That tells you that you have a problem with epigenetics, which actually controls the major gene in the hippocampus right area which can take this. They also then treated with inhibitors that will overcome this problem with methylation. They'll unmethylate them, and in fact they see that now the babies are better. Okay, so that's the evidence. Now you have a functional evidence to support that yes, this was important, right? It was just not that okay, we observed this, we observed this. It was like, okay, I now modify the observation and I can revert the phenotype, right? That is holy grail in most scientific experiments. If you can do that, then you have actually proven to the world that the observation you made was actually functional in nature. Okay, cool. I speak too fast. We have 15 minutes to finish. Okay, yes. Yes, learning leads to memories, but learning is learning. You learned a task, right? I say, okay, go on, fetch this bottle every single time. We'll do it right now. But 5 days later, if I asked you to do the same task, that is in your memory. If you learned and memorized it, right? You had a natural memory of that would be memory. So learning will lead to memory, but learning do not necessarily need to result in memory, right? एग्जाम के पहले आपने पढ़ लिया कि मैं ये सही होता है, इससे ये होता है, इससे होता है, वहां पे जाके लिख दिया, and next year I ask you the same question, you don't remember it, that's loss of memory. You learned it for that time, that's a short-term memory, then you forgot it in a long term, right? So there is no memory, right? So learning is not equivalent to memory. Learning leads to memory, but that's why you have to learn properly. Right? That's why we talk about not cramming and all of that, right? So there's a whole field of memory, my friend, that you can go read about. This short-term memory, long-term memory. That people have done lots of work in the field as to how the long-term memory is built, and that is why the Western education system actually is based on those experiments where people, and again, this is what I was telling you guys one day, right? That you memorize something, then you write it down. I told you to record yourself. I told, right, to do that, because then by the process of that, right, you change things in your brain at certain places, and I don't remember where, but there now you have created a memory of your learning. So you get the difference. There's a difference. But learning is obviously crucial for memory. You cannot memorize things that you don't learn, right? But learning is not dependent on memory. So learning step 1, memory step 2. At least that's my understanding, right? Cool? Break down things in different places. So this was beautiful, right? This has been observed in humans as well with a lot of associations. So this is a Harvard University's Center for Developing Child's pamphlet. Okay, that tells you that experiences early in life, while the brain is developing rapidly, because our brains are developing, we are still making new neurons, new things, right? The epigenetics, what you set it up as, is absolutely crucial, right? So therefore, that you are told to care for your baby in a nice way. Don't give them stress, right? Again, we know this, right? Children who are taken away from their parents and grow up in foster care actually have neural problems, right? People have done this kind of work to show that such kids actually have problems in their epigenetics of different sorts. Okay? So this has been well accepted that epigenetic changes early on in your life as the embryos develop or as the babies grow actually help you, make better behavior. That is equivalent to what we showed earlier, right? Because now you have better neuronal systems. Your neurons are functioning much better. Okay. All right. Go back to now neurodevelopmental disorders. Where were we? Neurodevelopmental disorder. Sorry, I'm actually sort of switching between back and forth, and apologize for that. But if you have any questions, ask me later. Okay. Yes. Epigenetic effects. Neurotransmit. I don't think there is any evidence for that. Yeah, probably not. Yeah, at least I don't know of any. Okay? दुनिया में सब कुछ हो सकता है, right? I don't know. I mean, हो सकता है. But देखो, the point is, what is neurotransmit? Right? What happens in neurotransmission? Chemicals go from one to another. If those chemicals impact any of these enzymes that put the histone marks or DNA mark or anything like that, then hoga. You have to do the experiment, right? What I can tell you is that yes, secreted metabolite from cell number 1 to the cell number 2, yes, can change epigenetics. Okay, so yes, neurotransmitter is a kind of chemical. It potentially can change. Have people studied it? I do not know. So you can go do your own research and see if you find it. Okay, it's an interesting area. We work on something where we look at a cancer cell. We isolate something called exosomes from cancer cells. So cancer cells and many cells in the body secrete these small vesicular organelles called exosomes. They have membrane, and they carry some of the DNA and some of the protein from the original cell, which secrete them. These cells, these exosomes, contain microRNAs, which you will hear about later. MicroRNAs obviously control gene transcription or translation, right? To change the level of proteins. They also carry a lot of epigenetic enzymes themselves. So we have a project in the lab, which is not published, where we show that the enzymes that are in the exosomes, so essentially secreted by cell A, when the exosome now is received by cell B, so if I take the exosome, put it on cell B, the cell B takes up the exosome, okay, and now cell B's epigenetics has changed. So that's best I can tell you, okay? But people have also demonstrated, of course, that there are different kinds of proteins, peptides, well, chemicals that impact the microenvironment of sorts. So our experiments were more focused on saying whether, if I don't worry about replication-based changes, so heritability, or if all my epigenetic changes are not due to cell replication, right? Can I just, in two somatic post-mitotic cells, I said something, did you guys hear that? Somatic post-mitotic cells, which is not going to replicate essentially, can two cells educate itself about the epigenetics, right? So we did this experiment for that purpose. But that is I think most similar to what you're asking. Okay, cool. So that can occur, I would say. But yeah, you can make a proposal about it, right? Write a proposal, make a hypothesis, propose experiments, and people will like it or not, and they'll give you funding or not to do the experiments. That's what to me, sure. I can't do anything. I can't fund your research, right? I can, of course, give you feedback. So yes, please. Thank you. Okay. All right. Do you get it? It's easy. Not easy, but at least I hope it's. So Fragile X syndrome, I just picked up two syndromes to tell you about, the neurodevelopmental syndrome, so you can understand what kind of epigenetic changes happen. Also because the slides are available for these easily. Okay. So Fragile X syndrome has a gene called FMR1, in which there are multiple repeats, a very unique kind of neurodevelopmental syndrome. So CGG motif gets expanded in this syndrome a lot. What happened to my previous slide? Or am I miss? Yeah. Okay, come on. जरा PDF है आपके पास वो जो मैंने भेजा वो जो आखिरी वाला PDF भेजा होगा ना आपको सर उसमें होगा शायद I had a picture here. I'm sorry I had to explain to you guys better, but of course I messed this up, right? So if he has a picture, I'll come back to that. There should be more than that here. Yeah, no, this is not correct. Okay, well, bottom line is just for you guys to know, in Fragile X syndrome, which has really bad मैंने आपको सही चीज भेजा है, उसमें क्या है पिक्चर दिखाइएगा एक बार। हाँ, ये आपने कौन सा लगाया है इसमें? ओ नहीं, यहाँ पे है तो। ओ वह क्या है, सॉरी सॉरी। Okay, fragile X syndrome is this. Okay, has a lot of problems with neuronal functions, right? So my slides were misarranged. I'm sorry. Okay, but that's uh, so there are intelligence issues in these people, these kids who are born with this. There are mental health problems, there are behavioral issues, and there are physical issues. Most of the neurodevelopmental syndromes also actually seem to change facial features and all, which and that's how in your society you recognize those kids. Actually, you'll see that. I mean, you most of the times you'll be able to tell that this guy has some problem because they have very abnormal features like either jaw issues or their eyes issues or the larger forehead, etc. I mean, you'd see this, right? Or nose. I mean, some facial features are also very obvious for whatever reason. So a large forehead in this case, a narrow face, and all that. Okay, so that's fragile X syndrome. This is an inherited disease. This is how kids look. You can see differences. Some things, right, from a paper. What happens in this case is commonly you have where was that? Before I say something wrong, right? Yeah, it's not wrong. Okay. This is caused by the CGG trinucleotide repeat expansion in the 5' region of FMR1. Usually you have 6 to 50 repeats, but in kids who are affected, you have over 200 CGG repeats, okay? And that's the genetics of it, but CG reminds you of methylation, right? So these kids also, there's a lot of methylation in this, and that is correlated. Also, there is also histone modification problem on this gene. This is a monogenic disease, again, with one gene, FMR1. There's also issues with FMR gene, right? So when FMR gene's levels are different, it also impacts MBD protein. So there is, loop, forward loop, etc. So these kids have a lot of methylation problems, bottom line, okay? That's caused by this. We talked about this. We're going to talk about, oh, that was my, I see. PDF हुआ था? नहीं, आप उसको निकालेंगे क्या? PDF को? अभी है इसमें? Sorry, there was one picture I want to show you of the, to introduce you to, नहीं है इसमें. हाँ. There's a syndrome called Rett syndrome, which I mentioned earlier. It's actually a big problem in Western world. I don't know if that happens a lot in India. Actually, I should know better. That happens because of only mutations in MEK2. That is our methyl binding protein. So people have been studying years and years and years, and we have not still figured out a solution. Very interesting. One monogenic disease. So most of these diseases actually don't have a solution. Okay, what you're seeing. So there is an unexplored world, is an unmet need, clinical unmet need, as we like to put things on. Okay, clinical unmet needs need to be met. So those are the ones which cause you, which research, right? So in any grant proposal I write, I say, here is a clear unmet clinical need, and we need money to solve this disease. That's my first sentence, right? That's how everybody does it. PDF. Second one. Third one. Oh yeah, that's PDF. Okay, let's see if I get to this. I don't think I can teach more than 6 days ever again. This was already too much on my brain. Okay, this is the slide. Did you guys see this? Rett syndrome. Rett syndrome has a lot of defects on the muscles and the movements, and I think they die at some point. These kids, they don't live full life. The muscle weakness, there's difficulty swallowing, difficulty speaking, and obviously brain issues, breathing issues, heartbeat regulators, all happen due to one mutation, MECP2. Okay, that's the gene. MECP2 is a methyl binding protein. It has role in neuronal maturation, like we discussed earlier, like for DNB3A3B, and it impacts multiple cell signaling pathways by ribosomal bonogenesis. Obviously, all the transcriptional things that we have looked at make sense. Very, very strong protein, very important proteins. Lots of functions, right? it, it also happens in autism spectrum disorders, and this particular disorder called fetal alcohol spectrum disorders. Okay, that's expression is down. Normally, this has multiple functions. It will recruit co-repressors such as ENCODE 1, 2. I will use the word co-repressors. Did you guys understand what does that mean? Right? No. So some proteins, the translation factors, actually will repress certain genes by not really recruiting activators, but actually recruiting repressors. There are several specific repressors in our body, in our cells, I'm sorry, like NCORE is one. CoREST is a big one, COREST, CoREST. It's actually found very highly expressed in neuronal systems, okay? And NCORE is its partner, it's another one of its kind. These recruit several histone modifying complexes, okay, such as LSD1, the histone demethylase for H3K4 dimethylation, histone deacetylases, HDACs, and chromatin remodelers. Some chromatin remodelers are repressors, not activators, because they assemble the nucleosomes together, so they'll repress. Okay, so MECP2 is actually a repressor in most part. Okay? It recruits co-repressors. And that can change the gene expression of multiple things. Okay, it also has, I think, role in 3D chromatin organizations, etc. Very, very well studied. This has been studied a lot because of the Rett syndrome problem. Okay, Rett syndrome is a devastating, kid syndrome in the kids. Okay, so it says in the bottom, in the disease setting, what happens? There are mutations like this, which are shown on the bottom. It allows the methyl DNA binding protein to bind less to the methylated DNA. Of course, it's a very good methyl binding protein. I mean, binds CpG. Okay. And then, as we have learned, when you bind CpG, you need to repress the gene, right? Because it's a methylation normally causes repression, normally in the promoters. So MECP2 will bind, and that should help you understand why I talked about co-repressors. If it was not clear, it will build co-repressors and silence the gene, because methylation is supposed to do that. So MECP2's role is to bridge that gap, right? It has domain to bind and recruit chromatin remodelers, recruit other histone modifiers that will put the repressive marks on and repress the gene transcription. You see the value of what we were learning, right? Okay. Okay, so it will change how the nucleus, etc., and all of that. So that's what the slide is telling you. Cool? MECP2, interesting, this is a Cell paper from couple of years ago, actually binds to 5-hydroxymethylcytosine. I told you guys 5-hydroxymethylcytosine. Did you guys remember what was it? No? None of you remember 5-hydroxymethylcytosine? No? So when I was explaining to you guys active DNA demethylation, this is the major process how active DNA demethylation happens. Your 5-methylcytosine gets converted to 5-hydroxymethylcytosine by enzymes called TET3, okay? Okay, let's go here. TET here. You see TET, TET1, TET. TET proteins are hydroxylases that will convert the 5-methylcytosine into 5-hydroxymethylcytosine. Okay, cool. So 5-hydroxymethylcytosine, obviously, once methylation is gone, it is trying to make the genes active. So this was an interesting paper that came out that talked about how MECP2 binds to 5-HMC enriched active genes. So MECP2 doesn't just have a role in repression, but it also has some role in gene activation or keeping some of the genes in a specific conformation. Okay. And then they showed that changes a lot of accessible chromatin in the nervous system. But the very important thing in this was that this did not correlate well with the gene expression. In fact, one of my PhD students worked also in another lab, a very famous lab, who does Zogby's lab in Baylor College of Medicine, which is next door to MD Anderson in Houston. And they showed that there is very little, if any, change in gene expression in this disease, in the brains of these people, of these kids, which is so funny, right? It's not all about what I was trying to get to you. But there's a lot of change in epigenetic modifiers. So either we are not able to capture the gene expression changes, they're too transient in nature. So if in a static world, when we look specifically at that time, we're not able to capture those changes in gene expression, or they are less consequential but more consequential things are changes in histone modification patterns. Okay, cool. Early on in day one, I talked to you guys about something called chromatin damage signaling. Did I? Do you guys remember any of that? So basically, what do I mean by that? I told you there's a DNA damage. DNA damage is when DNA sequence is not perfect. Chromatin damage, if you will, I'm coining the term now for you, is something where chromatin is not how it should be. Okay? So chromatin damage or reprogramming, as we call it a lot of times, occurs a lot. But can chromatin change, just a change in histone modification at this place to this place, could that be read by a cell? Not in terms of a gene transcription, but also just as such, could it initiate a cell signaling event? You get it, what I'm getting? Okay, that's a problem that is unsolved. That's something I think about a lot. Okay? How does that occur? Can cell really read a nucleosome that is not in its right modified state? Do you get it? So it should be, let's say, methylated at 5 positions, but it's not, right? So it will, what will it do? The proteins that read that mark now will be free floating, right? Because they're not able to find its substrate, right? Okay, let's take a step back. I guess I told you guys about a disease called MPNST early on, malignant peripheral nerve sheath tumor. Since we are studying neuroepigenetics, it's very important. It's a disease of peripheral nerves. It's a cancer of peripheral nerves. Okay. In that I told you a guy, a British actor who looked really weird, right? Remember? Right? That guy had a gene mutation in a gene called NF1. And I told you next to that gene in the genome is a gene called SUSY12. SUSY12 is part of the polycomb repressive complex 2 that marks H3K27 trimethyl. In patients who have MPNST, 70% of them, where there is SUSY12 deletion, they do not have H3K27 trimethyl. They just don't. Their cells just don't have K27 trimethyl. And I said this that time, how cool is that? Because now, do you understand why that is so cool? Because you studied a lot of things in the last 5 days. That's the mechanism for gene repression in our euchromatin. So there are thousands and thousands of genes that should be repressed are not repressed anymore, right? Because there's no more K27 trimethyl in that particular cancer. Why am I talking about this? I'm talking about this to make you think that there is a chromatin damage in this particular cell, because it should have had K27 trimethyl. That mark is not able to read. It doesn't have a substrate, right? So now it's free floating in the nucleoplasm. What is nucleoplasm? So it is a nucleus, that's nucleoplasm, right? So it might initiate new kind of signaling by binding to random proteins, by just, whatever, right? Could that now lead us to identify a new pathway that people have not discovered yet? And then could that be my therapeutic target for patients with MPNST? Get it? That's a question that's unanswered. I have just posed how I think about, I just told you how I think about new research in my lab. Okay? To me, that's a cool problem. I'm like, okay, boom. 10 years ago, when my colleague who sees these patients, who's a surgeon, told me about the disease, and I didn't know about the disease until 10 years ago. She said, Kunal, do you know this disease? I was like, no. She told me about this, and she said, okay, I would like to work with you on this because I'm the epigenetics expert. She's the surgeon who sees these patients, and she feels frustrated. I can't do anything about these patients. Just like I told you earlier, right? I feel frustrated. I couldn't do anything about my father, couldn't do anything about the girl, right?